Okay, welcome back. We're almost near the end. Today we're going to continue our discussion on the cardiovascular system, and then we're going to move into the immune system, which are related to one another. So we left off, we were talking about the heart and circulation, blood pressure, that kind of thing. And today what we're going to talk about is the delivery then of oxygen and the removal of CO2 to the different tissues. So what we end up having is we have our four-chambered heart, as you see here, and it's pumping blood to, say, all your tissues in your body, like your brain and your legs and your liver and all that kind of stuff. And as it delivers the nutrients there and the oxygen, it picks up waste products, brings that back to the heart, and then that pumps it to the lungs, and things like CO2 then get removed from the body. That's how it works. So in this case, we're talking about loading and unloading of oxygen and carbon dioxide. So first, what you're probably familiar with in vertebrates, let me grab my pen here. I think I don't know that I selected my pen. Anyway, there we go. So when we talk about vertebrate animals such as us, we have a protein called hemoglobin, and other animals, arthropods and mollusks, have a similar protein called hemocyanin, similar in their function, but these proteins are used to transport oxygen primarily to the tissues of the body. And so what ends up happening is oxygen's obviously transported to the cells of the body, and then CO2 is removed. And so what you see here is like what we call the percent saturation of hemoglobin in different parts of the body. So in the lungs, for example, the blood going to the lungs becomes 100% saturated. And then if you're exercising and you're working out, the muscles that are, under a lot of duress, if you will, the oxygen levels are low in those muscles as they use them up, as they use up the oxygen. So I think I might have mentioned this before about, the red and the blue that they show you. If you're just sitting there doing nothing, you're still at around 70%, 80%, so somewhere in here. So your blood is saturated throughout your body with oxygen, at least in terms of what it can hold. Now, obviously, what they mean by percent saturation is not that there's 100% oxygen in the hemoglobin, or it's not 100% oxygen. It means it's 100% of what it could carry. So there's a certain amount of oxygen that can bind to hemoglobin, and once you get to that level, you can't bind anymore. That's at 100% saturation. Now, there's this thing called the Bohr shift, though, which is kind of interesting. So it turns out that proteins like hemoglobin, probably hemocyanin as well, at different pH levels, their affinity to hold on to oxygen changes. So as the pH level drops, and again, that means there's more hydrogen ions and it's more acidic, the ability for the hemoglobin to hold on to the oxygen starts to diminish. And so what's useful about that kind of reaction is that as you're exercising or using up oxygen or doing cell respiration in certain cells, it starts to produce more carbon dioxide as a waste product, and then that allows more oxygen to be delivered to those particular cells, meaning that, like, if you're running, for example, then as your blood's circulating, you end up delivering more oxygen to your leg muscles because the oxygen is more likely to come off wherever you have a lower pH, which is what happens in exercising muscles. Now, CO2 can be transported in several different kinds of ways. About 7% of it is transported directly in the blood as carbon dioxide, as a gas. 23% of it are attached to the actual hemoglobin, but most of it, 70%, comes in the form of bicarbonate. So I'll go back to this to show you. So here's the cell right here. Here's my CO2. My CO2 comes in, and then it can undergo several things. It can react with the water and become carbonic acid and then break down into bicarbonate, like you see there, HCO3, or it can stay as CO2, as you see there. So it can travel in different forms as it reacts with other molecules. So most of it is traveling as bicarbonate. And I lost my pen. You ever lose your pen before? There we go. Okay. Now, one animal that's of interest, or one kind of, let's say, group of animals that are of interest, there are these deep diving mammals that we learned a great deal about in terms of how a cardiovascular system works and operates. So we have these deep diving mammals, and there's several of them. A Weddell seal, which is I think what this one is here, a Weddell seal. There's a couple. There's Weddell's seal. That's one. There's the elephant seal. That's another one. And then there's a whale that does this. It's a deep diving, very deep diving whale. I don't know if it's a sperm whale or, I'll have to look that one up. It's not coming to me now. Anyway, there's three, and they kind of have, they kind of argue over which is the best diving of these animals. They dive and can stay underwater. Like typically a human, maybe you can hold your breath for a minute or two. And if you practice and get good at it, maybe you do 2 minutes, maybe you do 3 minutes. But these seals, an elephant seal and this one whale, might be a sperm whale. Can't remember. Sperm whale doesn't seem right, but I can't remember. Anyway, seems like it's a toothed whale though. Anyway, they can hold their breath for a long time, like 20, 30 minutes, an hour, long periods of time. And that's of interest. Like, how does a mammal hold their breath so long? And so one of the things you might think, right away at least is, well, in us, we have lungs. And so when we hold our breath, we take in a big breath of air and we hold it, and then we go under the water, which seems logical because, if you need oxygen and you get the oxygen by your lungs, you fill up your lungs, and that would make sense. But it turns out that's not at all what happens. And if you think about it, when you fill your air up with lungs are buoyant. It's hard to dive. These animals dive really, really deep. So what they found out is actually their lungs aren't all that significantly big or anything of that sort, but they do have a huge amount of blood, and they have a huge spleen, and they have lots of myoglobin, which is a protein that can store oxygen as well. And so what they do instead is they will saturate this large volume of, much larger volume of oxygen, or volume of blood that can then hold more oxygen. And then they do things like shunt a lot of blood away from certain parts of their body. It allows them to hold their breath for a long period of time. And what they do is they exhale actually before diving, because otherwise that pressurized gas gives you a condition called the bends. If you ever heard of that before, if you dive, my youngest son likes to do every kind of thing you can think of. Right now his thing is he wants to go diving, and he wants to build his own tank in his own regulator. He wants to buy one on Amazon for $5 and build one. And I usually, I try to help him with all his crazy ideas, because I was that way as a kid. But there's certain things like building your own thing to fly and diving you don't mess with. Because if you go diving and you breathe pressurized air at the wrong pressure, you can get the bends and you can die. Actually, it forms these gas bubbles that travel through your body, and it's not good. But he's still trying to make one. Anyway, welcome to my world. So anyway, there's that. They also will decrease their heart rate and oxygen consumption, so they stop. They slow their heart rate down. They have this highly saturated blood with lots of oxygen in it, and they reduce blood supply to those muscles that becomes anaerobic, and that's how they reduce oxygen consumption, which means that oxygen they have will last a lot longer. Okay, now a little bit about body defenses. Part of this I'll go through quick because I want to get to the detail parts. It's a little bit tricky. So when we talk about body defenses, we're talking about ways your body defends against some of the things we talked about before, like some of the pathogens we talked about before. And you have some certain defenses that your body's just born with, and we call that innate immunity, and we'll get to that in a second. Some of those are things like your skin and some internal defenses. And then where we really want to spend the most time is on acquired immunity. This is what your immune system develops, and I'll show you how that operates, and that's the interesting part. So first, let's just briefly talk about invertebrates. So invertebrates have, just by being born, say, an insect, there are certain characteristics they have, like this chitin. You notice with an I there, I might have mentioned this before. Chitin is it's like a complex carbohydrate structure that covers an insect's, like its skin in part, and that's different from the chitin with an O, which is the animal. But they have that in their intestines and along their skin and elsewhere, and the chitin prevents things from getting in on the inside. Also, they often will call, when I first learned it, innate is also sometimes called non-specific because it doesn't really target a particular pathogen. Okay. There are also specialized sacs of enzymes called lysosomes. Those have like low pHs and they're designed to kill things that get inside. And then there are hemocytes, which are specialized blood cells that result in phagocytosis. They eat small bacteria and things that get in there. And there's also antimicrobial proteins. Proteins the animal makes that will bind to foreign pathogens like a bacteria that's invading it and make it more difficult for it to attack. But these are all innate. They're born with it, and they're non-specific because they don't really recognize, say, E. coli or staph. They work kind of on anything. And then we have innate immunity also in vertebrates, like our skin, our digestive tract, respiratory tract, genitourinary tract, the acidity of our urine, for example. We have a very acidic stomach, so lots of bacteria can't survive in our stomach. Our skin, is a very good barrier for a lot of things. A lot of things can't get in through our skin, which is why burns or cuts can lead to really bad infections. We also have cells that have these bags of lysosomes that prevent things from getting in also. And then there's some invertebrates, at least, there's like phagocytic white blood cells that will eat any bacteria that they find. I'll talk about the inflammation response, I think. Well, maybe I won't. Anyway, the inflammatory response, where let's say you're stung by a bee or you get a splinter in you, and what ends up happening, you get swelling in that area, and white blood cells come and they kind of clean up that particular spot. That's part of the inflammatory response. Inflammation is one of those things that is it's useful on a small scale, but as it goes broad and you have lots of inflammation, that ends up becoming problematic. There are also antimicrobial proteins that will attack and attach to, again, non-specific bacteria or viruses. And there are natural killer cells that are designed to kill cells of yours that are damaged or infected, or they just kill things that aren't supposed to be there, but they don't recognize any particular species of them, which is what we get to when we get to the developed specific immune system. Okay, just a really quick about white blood cells. There I see coming up my inflammatory response. I thought I didn't have it, but anyway, we have a lot of different kinds of white blood cells. We have neutrophils, which are short-lived, but that's where you have the most of them. We have monocytes that'll form as macrophages. They'll eat up bacteria that aren't supposed to be there. These live a long time in lymphatic tissue. We have eosinophils that are not very common. They attack larger parasites like worms. So let's say you had a blood test done and you have a really high eosinophil count. That's an indication that you have a larger kind of maybe parasitic organism. It could mean other things too, but a worm infection is one of them. And then this is the inflammation response that I was telling you about. But like, let's say you poke yourself with a pin or whatever, and there's things on the pin, bacteria or whatever. And what happens is when your body gets some trauma, small trauma in a particular area, what happens is several of the cells in that area, such as basophils and mast cells, will release different signal chemicals. One being, say, for example, histamine. They release histamine and you get inflammation. You heard about people taking antihistamines that are designed to prevent that inflammation. But what happens is you can see this blood vessel here went from small to big. That allows, it expanded, it dilated, and other proteins are released like cytokines that will then pull in more white blood cells, and it will clean up the inflammation problem that you have there. You also have antimicrobial proteins, such as found in what's called the complement system. There are these 30 proteins or so that will, there's different ways they can do it, but they group together and they attack and attach and break down something that's not supposed to be there. Again, something that we're born with. It's a set of proteins that reacts in a certain way to damage organisms, pathogens that aren't supposed to be there. And interferon. Interferon is a protein that is produced by viral infected cells. And what interferon does, and we use this a lot in different kinds of treatment now for viral diseases, but what interferon does is one particular cell, like let's say you have this cell here, this one gets infected by a virus, it releases the interferon, and that signal protects this cell. This cell, because this cell's damaged, this one's not going to make it. The cells next to it, for example, when they get the interferon message, it allows them to make antiviral proteins to prevent the spread of the infection. Okay, now we're talking about natural killer cells now, and then we're going to use that to move in then to the specific immune system as we talk about the acquired immunity. So the first thing you need to know is that on all of your cells in your body, you have what are called these class 1 MHCs. MHC, do I see it up here? No. It stands for major, can't start with an H and get to major histocompatibility, I ran out of room, complex. Major histocompatibility complex. And there are different types. So on all your cells, you're supposed to have a class 1 MHC. That's how your immune system recognizes your own cells. Now, if that class 1 MHC is missing, or if it's messed up, it's produced in a way that's kind of damaged in a particular way, these natural killer cells will release chemicals to destroy it. They'll kill your own cell if it's lacking this class 1 MHC, or if the class 1 MHC is somehow damaged on it. Now, that is exactly why, for example, when they do, say, an organ transplant and they're talking about finding a match and all those kind of things, they need to find a very, very close matching group of class 1 MHCs that'll match the patient. And it's very hard to get that exactly right, so they have to put you on some kind of immune suppressor so that your body doesn't attack it. So it's kind of a little bit of both of those. It's finding the right match with the right class 1 MHC, and then it's also putting them on immune suppressors to kind of counteract how close of a match you didn't quite get on that. Okay, now the next thing we'll talk about, which is related to the class 1 MHC, is the idea of acquired immunity. And just like it sounds, acquired immunity is something that you acquire. And in vertebrates, we use another kind of white blood cell that I don't think we've talked about yet, called lymphocytes. And lymphocytes come in two forms. There are B lymphocytes and there are T lymphocytes. And B lymphocytes, so first of all, they're all made in your bone marrow. So when they talk about a bone marrow transplant, the reason they're attempting to do that is they're trying to replace the stem cells that would make your blood cells. So a bone marrow transplant, the sort of condition behind it, or the idea behind it, is there's something about your bone marrow at a very basic level that you can't make the right kind of blood cells, and they're damaged in one way or another. So they try to put in new bone marrow that would be able to produce these cells. And they can come in two types at least when we talk about them. So they all start off in your bone marrow, in your arms and your legs and your hips and that kind of thing. And there are B cells and there are T cells. Now the B cells are made and they mature in the bone marrow, meaning they stay there and they come out mature that way, or they're a stem cell and they come out as a B cell ready to go. And those make antibodies. And antibodies are, if you know about COVID-19 right now, hey, how about that? COVID-19, what we're really testing for a lot of times is we're testing for the antibodies. The antibodies are not the COVID-19, but they're your body's response that it's seen the COVID-19. So if you test somebody and they test positive for the antibodies, doesn't mean they're sick. It means their immune system has come in contact with it. And antibody testing is extremely common. It's pretty cheap. It's easy to do. It doesn't tell you if you're sick or not, though. It just tells you your immune system has seen it. Okay? That's what B cells do. Now, T cells, on the other hand, they're made in the bone marrow, but then they have to mature by traveling in the bloodstream to the thymus, which sits right above your heart. It's a gland right above your heart. And so they mature there and they become T cells. And T cells don't make antibodies, but what T cells do that are different is they tend to attack cells that are your own cells that are infected with something. So B cells can attack from a long distance, but T cells can't. T cells have to come in direct contact, and they're both important in different ways. Now, normally what happens is the way your immune system gets triggered or it fires up is there's an antigen. An antigen is a particular chemical that your body's responding to, and it's usually something that's on the bacteria or the virus. So there can be different versions of it, and those are often called epitopes or antigenic determinants. And what you see is, like on this antigen here, there are 3 different kinds of chemical attaching sites, antigenic determinants, and they're going to bind to that as a response. Here you have antibodies binding to it. Okay. Now, those antibodies or those antigens binding to the antibodies can do a bunch of different things. So here, for example, you have, let's say you have a virus. Let's say this is COVID-19. Okay. So here you've been infected with COVID-19. If you are infected with the COVID-19 virus, the antibodies can attach to that and make it so that that virus can't attach to other lung cells and that kind of thing. So we often test for many things, including COVID-19, using the antibody test because antibodies are all over your blood. The one that they got to stick down your nose that they're swabbing for is a direct test. They're looking for the virus itself. They're testing for the antigen, and that gets confused a lot. So there's two very broad kinds of testing. There's testing for the antigen, which would be like testing for COVID-19 directly for the virus, which is harder to do. That's why you got to, and it depends on the virus. In this particular one, you got to jam that thing down in someone's throat. I think they found a newer one that can be done with saliva, but then you wonder if it's just as good, and we don't know. But if you go on Facebook, it turns out everybody there that has read 4 or 5 articles is an expert on it already. And that's a joke because people spend 20 years learning something. And I will tell you, COVID-19 this thing's new. This thing's 4 months old. We're going to know everything about COVID-19 in like 5 years. We're not going to know about it in 3 months. So all your social media, Facebook friends and all that, nobody knows everything about it, unfortunately. That's just how the world works. Unless it came from outer space or something weird. Science doesn't work that way here. Okay? So anyway, virus testing, direct testing is like sticking the swab down in there. But most of the time what we test for in almost every kind of case, like when we test for HIV or we do a quick COVID-19 test where they don't jam the thing down your nose and they do it from a blood test, they're checking for antibodies. Okay, antibodies, because those are easy to test for because they're all over the place. Now, doesn't mean you have it, though. It means you were exposed to it. But it's a cheap and much faster mechanism. And it's usually very, very accurate. But as we've seen, there are sometimes tests that aren't as accurate as others. Anyway, the antibodies will attach to the antigen. The antigen's on the foreign invader, the thing that's not supposed to be there. The antibodies are one of the things that can attach to that antigen. Now, normally what happens is we have our B cells and we have our T cells, and they have different sort of receptor sites. Okay, so this is my B cell receptor, and this is my T cell receptor. We'll go into more detail on those in a minute. You don't need to know about the heavy chain and how don't worry about that. So that's my B cell receptor, and then my T cell receptor. Whoops, wrong button. Okay, no, don't do that. There we go. Okay, and there's my T cell. We don't need to talk about it. We already did that. Okay, now let's go back to this class 1 MHC once again. Okay, remember a class 1 MHC is something that is supposed to be on all your cells, right? A class 1 MHC is supposed to be there. So normally under normal circumstances, the class 1 MHC is produced and it's on your cells, and it's how your body recognizes its own cells. Okay. Now, if your cell gets infected with a virus or it gets damaged and it has cancer, for example, one particular cell, it will start either not making, or it'll make that class 1 MHC a little bit different. So it ends up looking a little bit strange. And if the class 1 MHC doesn't look right, it'll activate cytotoxic T cells that'll kill that thing. And we'll get to that in a second. Okay. Now, there's also what's called a class 2 MHC. And a class 2 MHC is normally not present. So what ends up happening is you have certain cells, and often we call them antigen presenting cells because they do exactly that. They present antigens from things that they've come across in the environment of your body. So what happens with a class 2 MHC is it'll work with both cytotoxic T cells and helper T cells. But what it does is there's some kind of microbe. It could be a COVID-19 virus. It could be a bacteria. But this antigen presenting cell binds to it. It engulfs it and it breaks it down into pieces. And then it builds this class 2 MHC. and it puts some of that antigen on with it. So here's my class 2 MHC. So you can see that the class 2 MHC has my class 2 MHC part, but it also has part of the fragment from that organism, and that's going to bind with something like a helper T cell. Now, a helper T cell isn't going to kill this cell. It's going to trigger a response that communicates with other cells. Whereas what happens with the class 1 MHC, the cytotoxic T cell is going to kill this thing directly. Okay? Now, so you have your antigen, and again, the antigen is the invader. It's the thing coming in. So let's say you've got COVID-19. Someone sneezes in your face because you're at a protest or a riot or whatever is going on in the world. Like, who would have thought that we'd be in a world? And don't forget, one of my friends said, don't forget the killer hornets or whatever that is. I guess they found a couple. It seems like that maybe not is as big of a deal as we originally thought. But I've forgotten about those. But anyway, antigen, COVID-19, or some kind of bacteria, it enters your body, it comes into your lungs or whatever it is, and that antigen can do several things. It can directly stimulate B cells. Those B cells can start producing plasma cells. They'll replicate and they'll make a bunch of antibodies. They'll start fighting it. But an antigen presenting cell can also eat up that antigen and then present it to helper T cells, and then activate cytotoxic T cells and memory T cells, so that if you see that thing again, you generally don't get it on the second response. And the memory B cells then can, or the helper T cells then can help activate memory B cells and cytotoxic T cells, or they can also, that cytotoxic T cell can become an active cytotoxic T cell. So often we draw these two branches of the not innate, acquired immune system in two branches. We call this one humoral immunity, and this is the B cell one because it's in your blood, and this one's cell-mediated. These are the T cells, and we draw them separate like that just as we learn them. So we have B cells that are making antibodies. We have T cells that don't make antibodies, but they attack things differently. So in a way, it looks like there's two separate things, but you see right all along here, there's all this interaction between them. So the B cells and the T cells work together in reality, although they can do different jobs. As a general rule, B cells, for example, which produce antibodies, can attack things that are anywhere in your body. Anywhere the blood travels through your body, the antibodies can reach. Whereas I don't know if I said that right, but B cells travel, B cells produce antibodies that travel through your blood and can attack things far away. T cells, on the other hand, have to come into contact. So if your cell has cancer, it has a virus in it, B cells can't get at that cell directly. They can't kill it because the antibodies can't kill one of your own cells per se that has the virus in it. It needs instead the cytotoxic T cell. So there's a slight difference like you see there. Okay. Now, normally the immune response when you first pick something up, it's slow. Takes 10 to 17 days from the initial exposure to an antigen, and then you start producing B cells and T cells, and then the secondary response is usually much quicker. Now, you might look at this and realize, like, wait, COVID-19, if you get infected with it, it seems like you get really sick in 2 weeks. That's just a coincidence. So other things take a lot longer. Some things take a shorter amount of time. But as a general rule, the primary immune response for most people when they first see an antigen for their body to make antibodies to it is 10 to 17 days. But if you've already been exposed to it, usually it's much quicker. So if I show you sort of this graph here, let's say I get exposed to COVID-19 right here. Okay, so first it takes a while for my body to make antigens to it, but in 3 weeks, if I'm out again, assuming everything is working like we think it normally does, and I come across COVID-19 again, what happens is I make this really quick response to it. So you're likely in many cases to maybe get sick the first time from something, but quite often you don't get sick the second time because you have a much faster and much more aggressive response the second time. Now, these things vary, so it depends on the pathogen and how it changes over time and your own immune system. So that is all sort of a softball sort of view of it. Now, what I want to also point out is, like, let's say you're exposed to COVID-19 and you had that particular response, and then next week you go, oh, I was already sick, so I don't have to worry, my friend has influenza, so I'm going to go over there. Well, that's different. So the fact that your immune system saw COVID-19 on Monday, and then 4 weeks later, you're going to go visit your friend that has influenza, which is a different virus, it responds as if it's brand new. So you can make new antibodies to each kind of thing. But the advantage of it is you can tune it to being exact to one particular kind of problem that you have. So first, we have humoral immunity. And humoral immunity involves B cells. Like I said before, B cells produce antibodies, and they don't have to be next to the antigen to attack it because they can be in the blood and they can travel around. Then we have also cell-mediated immunity, which are T cells. And again, those have to be next to the antigen to attack it. Okay, now the next part here is what we're going to see is how the helper T cells work in response to almost any kind of pathogen. Okay, so helper T cells are really important. It's one of the reasons why HIV is so terrible, is because it takes out these T cells which function to sort of help all the other cells. Now, normally what happens is, we talked about the class 2 MHC, right? Class 2 MHC. What's going to happen with a class 2 MHC is it's going to bind to a helper T cell that's going to have its own receptor on it called a CD4 receptor. I always remember 4 times 2 is 8 and 8 times 1 is 8. And so if you remember 8 all the time, it makes it easier for me, but we'll see. Sometimes it works for you and sometimes it doesn't. It's there, it's like Todd Newberry said, you can either do it or you don't. That's some wisdom there. That's 20 years ago and I'm still remembering. That guy's a genius. Anyway, Todd Newberry, he's the famous tunicate guy. Anyway, here we have my bacteria. My antigen presenting cell tears that thing apart and it presents it, right? It takes my class 2 MHC, it takes some of the pathogen and it produces it together, and my helper T cell with a CD4 receptor, the CD4 receptor binds to the class 2 MHC, and that helper T cell then releases a variety of kinds of cytokines such as interleukins. Interleukins are a big group of chemical signals, and interleukins then travel through the blood and they activate B cells and cytotoxic T cells, and those then try to kill that particular bacteria in their various ways. Okay, now in a cell-mediated response, what you're doing is we're using cytotoxic T cells, but what we're really trying to do here is a little bit different. We're binding to a damaged or missing class 1 MHC. So let's say this is your cell here and you've been smoking your whole life or doing something bad or eating nothing but hot dogs, something that could maybe give you cancer, whatever that might be. And so this cell over time gets damaged, and as it gets damaged in a way where it becomes cancerous, it starts to produce a class 1 MHC that isn't perfect. It's not exactly the same. It doesn't look like it's supposed to. And what happens there is, in your body, these cytotoxic T cells come along and they bind to class 1 MHCs using a CD8 receptor. Okay, so normally a perfectly functioning class 1 MHC won't bind to a CD8 receptor. So this one is one that's damaged. It looks wrong or it's missing. So what the CD8 receptor does is when it binds to it and it recognizes the match isn't perfect, it releases proteins such as perforin, and that's a protein that will put a hole in this cell and will kill it. So what it does is it causes, it stops, if you will, homeostasis. So you have this cell, it's got a membrane, and it prevents things from just moving in and out freely from the cell. It controls, it regulates what happens in the cell. When you put a hole in the cell, it becomes leaky and you end up killing that cell and it dies, which is kind of a good thing in a way. So if your cell has cancer in it, rather than replicate that and let that spread, your immune system is designed to kill your own cell that might have cancer in it. The problem is with something like with HIV, the cytotoxic T cells are killing your own T cells, and so that wipes out your cell-mediated part of your immunity. Okay, and again, B cells, remember, they produce antibodies. That's the key there, and they can live a long time. So you can produce these memory B cells, and that's why you have that quick reaction later on. Now, there are a bunch of different kinds of antibodies, and I'm only going to tell you about two of them, IgGs and IgAs. IgGs are the most common, most abundant one, and these can cross the placenta. So if a woman is pregnant and she's got a baby growing inside of her, the baby has, it's missing, it doesn't have a very good immune system before the baby is born. So the baby gets some immunity from mom because these IgGs can cross the placenta, so it somewhat protects the baby. Baby isn't protected against all things, but that helps. Now, once the baby's born, though, the IgGs, they don't last very long. So whatever the baby's born with, it has those proteins in it for a short period of time, but doesn't have the defense, really, doesn't have the ability to produce the IgGs. So babies then can get some immunity from mom via breast milk because IgAs are present in breast milk, and that can protect the baby for a certain amount of time as well, because the baby, he or she, is getting mom's IgAs. Okay. Now, ultimately, though, what ends up happening is giving the baby the antibodies helps protect the baby right then and there, but the baby can't take care of itself later on. So we have sort of two kinds of ways you become immune to something, and one's called active immunity and one's called passive immunity. So in active immunity, your body can take care of itself in a sense. Active immunity is where your immune system, when it comes across the antigen, it can respond on its own. It has its ability to do it. And you can get that two ways. You can get the disease, whatever that is, and then if you live from it, you now can make the antibodies. And the idea is that once you've seen it, you can make antibodies and you won't get sick again. Or you can get immunizations or vaccines. You can be immunized. And that's the big talk right now. That's the big deal right now, is trying to make a immunization for this COVID-19. And then once the riots and all that are over, everything can go back to normal. We'll all be perfectly happy. But we got a lot, unfortunately, we got a long way to go. So when I was a kid, for example, we didn't have a vaccine at that time for chickenpox. So when you were a kid, if your friend Bobby or whatever had chickenpox, your parents brought you over there to play with Bobby, and you had to get the disease. I mean, it sounds like archaic almost. It sounds like, I lived in, the Middle Ages or something weird like that. But that's how they did it. It's crazy thinking about it now. Now, usually I ask a class of students, how many of you have had chickenpox? Maybe a few of you. I had it, not fun, not good, but you're immunized. My kids are immunized against it because now they have a vaccine for it. So you either get the disease or you get immunized for it, is the general rule. Okay. Now, then we have a couple things that happen, like, for example, with the flu. There are certain things that change a lot. Chickenpox doesn't change all that much. Stays kind of the same. But there are some things like the flu, which are constantly changing. So sometimes you can get vaccinated for something once in your life. Sometimes you need it 2 or 3 times. There are different versions of it. The flu is one of those where they're always working on it. And unfortunately, it looks like COVID-19 might be one of those that we have to keep after. It'll be changing perhaps, and maybe you'll have to get vaccinated for it all the time when there is a vaccine. And that's the big problem. People don't understand that. Why isn't there a vaccine? So the trick is, the problem is, here's the problem. Treatment is way easier than the vaccine. And here's why, at least here. Treatment, you got a sick person's sick, they're not in good shape and things are not going well, and you're trying new things out. And we know, like, for example, for COVID-19, these people are sick and a lot of people are dying. So treatments that you do, if the patient ends up dying, it's kind of hard to know, did they die from the treatment or were they going to die anyway? There's a lot more flexibility in trying new things out. Okay, for treatment. But for a vaccine, the problem is you have people that are healthy. You usually don't vaccinate sick people. You vaccinate someone that's healthy. So when you develop a vaccine, it has to be really, really tested very, very well over and over and over again, because if you develop a vaccine that actually makes people sick, there ends up being a lot of lawsuits and a lot of problems with that. And so it seems like it would be an easy thing to do. You would think, we just need a vaccine. Where is it? Why won't science save us? Because it takes a long time, unfortunately. And you get what you get, and you don't throw a fit. Miss Fitzharris, remember? So vaccines just take a long time. I think they said the fastest vaccine ever developed like 3 years or something like that. But it could be 3, 4, 5, 10 years, because you have to get it right, and that's how it works. So that's active immunity. Now there's also passive immunity, which is more kind of like treatment in a way, if you will. So passive immunity, you can get antibodies or antitoxins, whatever it is that you're getting. But passive immunity is like when you're pregnant or you're breastfeeding and you're getting antibodies, or if you get sick in one way, or you get poisoned by something. For a lot of things, we have antibodies for a particular snake venom, for example. They have antibodies for if you get bit by a snake or a spider, or so there are a lot of ways that if you are infected or you're injured, they can give you antibodies for a particular pathogen to sort of save you in the short term. Now, the problem with the antibodies, again, is you don't have the ability then to produce them later on. So ideally, you want antibodies to be able for your body to make them, but sometimes in a pinch, you don't have that option, and antibodies are the only way to go. Your immune system then can also kind of go kind of crazy, and it can do things like attack itself in one way or another, or it can overreact to things. People that have seasonal allergies, for example, or if you're allergic to bee stings, understand this concept. Sometimes the immune system will actually attack itself, and autoimmune diseases like lupus, MS, ALS, Lou Gehrig's disease, rheumatoid arthritis if you have it. So rheumatoid arthritis is different than, like, if you ran 20 miles a day and you kind of wore your joints out. Rheumatoid arthritis is like where your immune system starts attacking the joint. So you can get arthritis in different ways, but all of these are autoimmune diseases, which means your immune system sort of attacking itself. And then you can be actually born where your immune system just isn't very good. You can just have a lower immune system, or you can have SCIDS, which is severe combined immunodeficiency syndrome, which basically means you're kind of born without an immune system. And if you've ever seen John Travolta's Boy in the Bubble, and then there's other versions of it, but essentially you can't come in contact with antigens, or you can get sick and die from it. And then, of course, everybody knows about AIDS and the HIV virus. In this particular case, you have a virus that tends to get inside your body. It can go anywhere, but it tends to infect T cells, where it causes the most problems. And then those T cells get wiped out. You have no T cells, and then you end up getting, a lung infection or a kind of cancer or various things that end up killing you from that. Okay, now, just since we're mentioning HIV on our last slide here, there's different stages of how HIV is spreading and what it's doing in your body. So first, what ends up happening is if you look at the antigen itself, when you get infected, it replicates very, very quickly, but then it goes dormant and it sort of hides out. Okay, and in the very beginning, there's this slow response. It takes quite a while for your body to produce antibodies against it. So what ends up happening is, if you have a patient and you think they're exposed, were exposed to HIV, if you tested them like a week later, they're probably going to test negative, because most of the tests, unless you test for the antigen itself, but when they test for the antibodies like they often do, it's going to come out negative because they don't have the antibody yet. So it takes a while. Whoops, I'm on the wrong curve there. Where's my antibody? Here's my antibody curve, the red one. I guess I was kind of on it, right? So it takes a while before you have this high antibody concentration. So like on year 3 or 4, you're like, hey, let me do an HIV test. Well, by then, you're going to have a pretty consistently high amount of antibody. But the HIV kind of disappears. But as the HIV starts to spread and replicate, what ends up happening is it starts to infect T cells, and your T cell count drops. And once your T cell count drops, it takes out the cell-mediated immunity, and you lose that part of it. And then you're opened up to all kinds of issues after that. A lot of people are kind of getting wacky, saying, hey, these COVID-19 patients, they're dying from heart disease or the flu, and they're making the cases up. And I keep pointing out, what about HIV? How many people die from HIV? Because really, in the case of HIV and AIDS, you die from all kinds of different things, pneumonia, cancer, because the HIV itself is just wiping out your immune system. So COVID-19, for example, they have ways to test you to see that you are infected with COVID-19. You have these kind of unique set of symptoms that can vary that cause heart failure or pneumonia and those kind of things. So the idea that a COVID-19 patient died in a car accident and then they labeled them as a COVID-19 patient is sort of an imaginary thing that people make up. I mean, it could happen maybe, but you can't you can't get numbers that high by making up. You'd have to make up like everything. And that's unfortunate. But people fall for things like that all the time. So anyway, there we go. We're going to move on. And we have one more lecture. The immune system. No, we did the immune system. Starts with the nervous system.